Assalamualaikum and a very good evening. We are from Group Gene Stunner and today we'll be talking about genetic material. Genetic material refers to those material that are found inside the microbes that serves as a template for producing protein structures for their cell growth. Next.
Okay, next. In process of determining the genetic material, the first experiment is Griffith experiment by Friedrich Griffith in 1928. He studied bacteria of Streptococcus pneumoniae because there's only strain S3 and R2 used determining the develop vaccine for pneumonia. He isolated the bacteria from herd of the animal and ejected the mixed strains. In observation, a chemical component from virulent S cell transformed R cells to virulent S form. In conclusion, from one cell to another cell, a chemical substance is genetically transforming. Next.
Avery, Macleod, and McCarthy in 1944. They repeat and improved the Griffiths experiment using a purified cell extracts. They found that the protein and RNA is not a transforming factor, while the DNA is the transforming factor. In the conclusion from the Avery's experiments, they come up with the hypothesis that the genetic material of cell is either protein or nucleic acids. But the conclusion is transformation requires DNA, thus it is the genetic material of the cell.
And next is a Hershey and Chase experiment by Alfred Hershey and Martha Chase in 1952. Investigate bacteriophage. Do we only DNA and protein composed in it? So what he do, phage are protein code and DNA radio label, and it's injected into the bacteria to determine which one the genetic material. In observation for protein, no radioactivity into the cell. For DNA, radioactivity into the cell. In conclusion, the genetic material is identified are DNA, not the protein. Next.
For the next experiment, conducted by Hans Alfranco Conrad in 1955 to prove that is a genetic material. So he uses the tobacco mosaic virus, or we call it as TMV, to crisscross another virus to change its RNA rod and also the protein structure so that one has a different protein structure and a different RNA rod. From the observation, the TMV virus that affected the specimens shown that it only produces a TMV virus that are present at the experiment. So in conclusion, the experiment proved that the RNA is indeed a genetic material for the certain viruses.
So, in conclusions, the genetic material in fact that found in the bacteria is DNA, while the some viruses, the genetic material is RNA. That's all from us. Thank you.